Well, let's get started. So this was a very dense chapter. Probably read it over a month ago. So I tried to pick over what I thought was important, and I think really the big concepts are what's important. And then you can probably pull from the pictures, which I thought were pretty good in this chapter, what you're going to need to memorize, when it comes down to it. Okay, so B and T lymphocytes, which was the focus of this chapter, both start in the bone marrow, which is one of the central lymphoid tissues, and then the T cells go to the thymus, which is the other central lymphoid organ, where they mature. So the central lymphoid tissue is to be differentiated from the peripheral lymphoid tissue, which would be things like the spleen and lymph node and MALT, where the mature lymphocytes would reside, ready to fight infection. Okay, so first broadly we're going to talk about B cell development, which in broad strokes basically starts in the bone marrow and is mediated by interactions with things secreted by the bone marrow. A B cell is made and then it's checked to make sure it's not autoreactive, and if it's okay, then it's mature and it goes out into the periphery where it would encounter antigen, where a naive cell would go and encounter antigen, and then it would be activated in the B cell and can eventually become a plasma cell or a memory cell. Okay, so now we're going to go through this in a little bit more detail.
Did you start recording? Yeah, okay.
Did I?
Yeah, you did. Okay, see it now.
So B cells start, like all other cells in the bone marrow, with the hematopoietic stem cell, which then becomes a multi-potent progenitor cell through different signals, then to a common lymphoid progenitor, then a pro-B cell, pre-B cell, finally an immature B cell where it leaves the bone marrow, and a mature B cell is the last stage before encounter with an antigen. A mature B cell is potentially still naive. It's naive until it encounters an antigen, even though it's mature. So this is the first great picture that I think is worth knowing fairly well. It's on page 278. Again, it's just what I mentioned, that it starts as a hematopoietic stem cell, becomes a multipotent progenitor, then onto a common lymphocyte progenitor, if it's going to become a BT or NK, and then a pro, then pre-B cell, and finally a B cell. Okay, so what helps it progress through these different stages? Well, the stromal cells in the bone marrow are probably what helps everything progress, because they're what's going to secrete the signals that are going to be detected by the cell and is going to help that cell decide what it is going to become or what it should be doing at a certain point. So in that second stage, that multipotent progenitor cell, a lot of it is dependent on this FLT3 interaction. And then as it becomes a common lymphoid progenitor, then IL-7 becomes more important. So IL-7 getting secreted from the stromal cell, interacting with IL-7 receptor. So it's a pro-B cell when it starts expressing KIT. So KIT you probably hear in terms of mastocytosis. So KIT is an important receptor on lots of different blood cells. KIT is made by the c-KIT gene. So you've probably heard of c-KIT before. The actual receptor is called KIT, and its ligand is stem cell factor. So then eventually at the pre-B cell stage, an immature B cell receptor, which we'll talk about more in a minute, is expressed. And then finally, most of these receptors recede, some new and stay, and quintessential receptor on an immature B cell is the B cell receptor, so a fully formed and functional B cell receptor that is not autoreactive. Okay. So as this progresses through, for that B-cell receptor to be made, it happens one chain at a time. And as it turns out, it's the heavy chain that comes first. So the heavy chain is the one that has both V, D, and J. The light chain has just a VJ. So at the early pro-B cell stage, D and J start rearranging. Because remember, DJ comes before VJ. Disc jockey became before video jockey. So the DJ rearrangement occurs first. And then second, at the late pro-B cell stage, V is going to rearrange with DJ. Once that's happened, you can actually get a pre-B cell receptor that is made up of the heavy chain that has been fully formed within a surrogate light chain. Now, once this happens, this is called a pre-B cell, right? For this to progress, this actually has to be able to signal and send a signal through. So one of the important components of signaling, right, is BTK, or Bruton's tyrosine kinase. So if you don't have a functional Bruton's tyrosine kinase, you arrest here at the pre-B cell stage. If you see a patient who has seemingly Bruton's but is a girl, well, they're also autosomal recessive forms of this. Particular, this, I believe it's called mutaine deficiency or something like that. So it's the same concept, that this pre-B cell receptor has not formed correctly and cannot signal through. So it's still going to rest at that pre-B cell stage and act a lot like Bruton's tyrosine kinase, but it could be a girl or a boy. So after this is successfully signaled, then gene rearrangement can reoccur in that light chain. We'll get a V to J rearrangement. And once that's happened, a fully functional IgM will come out to the cell surface. And that's considered an immature B cell. And then once IgD is on the outside, it's a mature B cell. So these were the proteins that were discussed and essential for signaling. I'll probably be familiar with most of these. FLT3 is the very first one where is essential in those early stem cells. Of course, KIT as it goes through those pro-B cell stage. We talked about IL-7 as it's getting to later. CD25 or the IL-2 receptor then kicks in. But there's a couple that are important as well in order for the B cell receptor to express, namely E2A and EBF, PAX5 and BSAP. Okay, so this, I don't know if this really says anything different that I haven't said at all, but this is figure 8.6 on page 281. And again, as a reminder that in this heavy chain, you have this V, D, and J. The DJ gets rearranged first, and then VDJ. On the light chain, it's just the VJ rearrangement that needs to occur. Of course, in B cells, and this is not covered in this chapter, keep in mind that a couple of things happen outside the bone marrow. So one being that this constant chain can rearrange, right? And then you can flip a switch and change from IgM to things like IgG or IgA. The other thing that can happen is after encounter with an antigen, you can get affinity maturation. So you can actually get further editing of this receptor to make it more specific for the antigen that's encountered. So on the light chain locus, in order to make this more likely to result in a productive immunoglobulin receptor, you can actually get a recombination, and if it's non-productive, then you can actually get a second or third recombination in order to try and rescue that, in order for that to happen. Okay. So again, any of the graphs that look like this in the chapter, I would probably pay pretty close attention to. This one, though, we've learned about most of this already this year, so hopefully this is review, and actually if you kind of put it into context, it makes sense, right? So if a rearrangement's occurring, well, what do you need? We've talked about this, right? RAG1 and RAG2 is essential. And then TDT, remember, this is where the N nucleotides are added. Well, these things are expressed at the time when these gene rearrangements are occurring. Of course, the surrogate light chain, we know as soon as that IgM is expressed, then that goes away. And then, of course, for signal transduction, remember that IgM doesn't transduce all the way to the nucleus, but it has some helpers that, like Ig alpha and Ig beta, they're going to help signal, and they're going to actually come up at a very early stage. Okay. So it is miraculous that B cells ever make it, but of course some do. The different stages where things can go wrong, of course, we get your early pro-B cell, you get DDJ rearrangement. If that is functional, great. If not, then you can get another rearrangement on the second chromosome. If that doesn't work, you've lost your cell, but you still have a chance. If that works, great. Then the light chain comes up, and then you rearrange kappa first, and then the second chromosome, then you arrange lambda, second chromosome. If none of those work, then you've lost your cell. If any of those work, then you've gone on to the next stage to see if that is something that can be let into the periphery. So any lymphocyte, once it's recombined, right, has a certain risk of being able to recognize the wrong things. And of course, the wrong thing would be yourself. You don't want to have an autoreactive lymphocyte. It can cause problems, as we know. Right? So different options are, it's perfectly fine. It doesn't seem to react to self, so it goes on in the periphery. Perfect. In B cells, if it is autoreactive, then it has one chance at rescue, in which is called receptor editing. So that receptor can be edited just a bit, and then one more try can occur to see if that is actually going to be okay. If after editing it's not self-autoreactive anymore, then it's off to the periphery. So when it's being tested and dies if it reacts to self, what kind of selection is that called? Positive versus negative? That would be a negative selecting event. So if the result is it's died, it's negated, it's negative selection, it's cast out. If it can be positively selected, so in the case of T cells, they're going to go positive and negative selection, right? So they have to recognize MHC because they're MHC restricted. If they do, then they're positively selected. They're allowed to live. So it's positive selection, right? B cells are not MHC restricted, so they don't have that step, but they still have this negative selection in that if they're autoreactive, they're selected against. All right. Moving on. Okay. So T cells, shouldn't say tolerance, start in the bone marrow, but they actually leave at a very early stage to the thymus, and you actually don't have T cells if you don't have a thymus. We know this because complete DeGeorge, if you're completely athymic, have absolutely no T cells whatsoever. In order to be a T cell, it had to have been a thymocyte at some point. So the precursors, though, start in the bone marrow, and then they are able to home to the thymus, and they start in the thymic cortex, and then they go on to the thymic medulla. What I think is interesting, actually, if you look at Dr. Markert's work at Duke, The thymus doesn't even have to be in any place particular. In fact, when she does a thymic transplant, it goes into the leg. It's these early cells are going to find it and home to the right spot no matter where it is.
Is there some early hypothesis that there's some differentiation of T cells in the gut?
Without being through the thymus at all? You know, I think there are thymic independent T cells, but I think they are strictly ones that are very low diversity. I actually thought gamma delta T cells were thymic independent, but this chapter says that they actually go through the thymus. But I do think you're right that we think there are some that even if you had, for athymic, you would be able to detect some of those really low diversity peripheral mucosal T cells. Okay, so in broad strokes, what happens, you get the T cell precursor, it arranges its gene, you get a functional T cell receptor, undergoes positive and negative selection. It goes out to the periphery, it finds antigen, and then is activated if it recognizes antigen. Okay, so the thymus is made up of an outer capsule and then the cortex on the outside, and the cortex is where the very immature thymocytes reside. And then before they end up back into the periphery, they're in the medulla. In the medulla, there's something called Hassel's corpuscles. I don't think we really know exactly what these are yet, but they may be stromal cells that have stopped expressing something called air, which we'll learn about in a little bit. It may kind of just be actually residual stromal cells, but I don't think we know quite yet. So T cells actually go through a separate stage in that they start as a double negative thymocyte, which means they're not expressing CD4 or CD8. And then they have a choice of either becoming a gamma delta T cell or an alpha beta T cell. If it's gamma delta, it is going to necessarily remain CD4, CD8 negative. So gamma delta cells do not have these co-receptors. But if it becomes an alpha beta T cell, which we think is the fate of most T cells, it's going to go through a stage where it's actually double positive thymocyte. So it's going to express both CD4 and CD8 first. It's going to, similar to the B cell, it's going to start with kind of a pre-T cell receptor, and then it's going to have a fully functioning alpha beta T cell receptor still in a double positive state, and then it's going to select either CD4 or CD8, and then you're going to have both T helper cells and cytotoxic T lymphocytes that are then going to be allowed to go out into the periphery. Similarly, different things are going to be upregulated at different times, and probably at least some of these are important. So one of the signals that seems to go through most of T cell development is something called Notch, one of the first things mentioned in the section. But KIT and stem cell factor interaction is also probably very important to T cells. So in this case, when it's going through its double negative stage, it's going to be differentiated into 4 double negative stages, or substages: DN1, DN2, DN3, and DN4. So in DN1, this adhesion molecule CD44, adhesion molecule CD44 is going to be expressed, and then it transitions to DN2 stage when CD25 is also expressed, that IL-2 receptor. The DN3 stage is when the pre-T cell receptor is expressed for the first time. So, of course, what's going to happen? Well, there's going to be gene rearrangement because you need that to get that first beta chain out there. Beta chain is going to be the first to be expressed. Beta chain is the one that has a D in addition to V and J. So you can get a D to J rearrangement first, and then a V to D, J rearrangement, and that's where you're going to get your pre-T cell receptor You're going to get some proliferation. You're going to lose both CD44 and CD25. Then you're going to become double positive. At this stage, all the mechanisms to allow for rearrangement have to be turned back on so you can rejoin the alpha chain, that V to J. And then you can have a fully functional T-cell receptor. Of course, at that point, you're going to lose one CD4 or CD8, and you're going to have a single positive T-cell. So this is basically saying the same thing, but just shows how the cells from the bone marrow come in at this cortical-medullary junction through the venules, and they start as these DN1 to DN2, DN3, where they're going to have that pre-T-cell receptor, DN4, and then they're going to be immature double positive thymocytes where they're going to develop that full receptor, and then eventually they're going to become single positive mature thymocytes or T cells, and they're going to be allowed back into the periphery. This is one point that had a long section, and I'm not sure you need to take much out of it, except that right, there's a competing interest in the cell to either become an alpha-beta T cell or a gamma-delta T cell. This seems to be mediated by this ERK signaling. So if there is, I'm going to mess this up. If there's strong ERK signaling, then it blocks beta, I believe, and becomes alpha gamma. Does that sound right? Right, strong ERK activation, double negative T cell induces expression of the transcription ID3, which promotes gamma delta. So strong ERK leads to gamma delta, weak ERK leads to alpha beta. So I had not learned this before, and I'm not sure there's much to take away from it except maybe that one point. Strong ERK, gamma delta, weak ERK. That seems like it'd be testable to me. So if it becomes gamma delta, remember, so I've talked about gamma delta T cells a little bit, but they have a very low diversity, and they're mostly found in the periphery where they're going to see a very limited number of things. So in kind of stages, they're going to go out to the skin, and then the uterus and lungs, spleen, lymph nodes. Right. Okay. The alpha beta T cells, to review, undergo just like the B cell receptor in that they start with the beta chain, which is analogous to the heavy chain. Heavy, it's got an extra segment. Maybe that's a way to remember it. VDJ, so you get DJ rearrangement first, and then VDJ. And then, of course, you also have to rearrange your alpha chain, and only then do you get an alpha beta. And, of course, you have to get expression of the rest of the T-cell receptor complex, which we've spoken of before, too, right? CD3, the zeta chains, and the co-receptor CD4 or CD8. But it's the alpha and beta chains that rearrange. Beta first, and then a pre-T alpha chain is going to hold it there until that alpha chain has time to rearrange and come out. Okay, so just as you would expect at the times when the chains are being made, so first the beta chain, then the alpha chain, RAG comes out, which is essential, TDP, which adds those new end nucleotide additions. And that surrogate light chain is going to go away as soon as you get a fully functional alpha chain. Signaling, of course, is important and has to occur both through this pre-T cell receptor and the fully functional T cell receptor in order for this T cell to survive. Although I don't know, so for an example, you could have a defect of ZAP70, which leads to a form of SCID. I don't know if you would have, That's actually a good question. What stage of, if you have thymocytes, or if that signal was important before? So BTK, I know they arrested the right pre-B cell stage. I can't tell you what it is for ZAP70, but again, you have to have the signal or you're not going to get T cells at all. It's not that you can have T cells that don't signal and then they just don't work. Right? You actually can't even make that cell if that signaling can't occur from deficiencies like ZAP70. Oh, I meant to bring this up. So there seems to be, right, some transcription factors that are important in making different T cells that I would probably know. And I'm not sure I'm going to be able to come up with all of them, but the transcription factor that's essential for Tregs, anybody know what it is? FOXP3, I think TH17 is something called ROR gamma tau.
What's it again?
ROR gamma tau or ROR gamma T. I think GATA3 is TH2. I don't remember what TH1 is. Do you remember what TH1 is, Dr. Barnes?
No.
Let me think for a second. I was thinking TH17. There's a section somewhere else in the book. I do think this is good, though. Something to know for your test. ROR gamma tau induction. Yeah, ROR gamma tau is TH17. Essential transcription factors. FOXP3, ROR gamma tau, TBET is TH1. And this is coming up actually in Chapter 9 next week, so maybe this is skipping ahead. But GATA3 is the essential transcription factor for TH2, TBET for TH1, ROR gamma tau for TH17, and FOXP3 for Tregs. Okay. Similar to the light chains, you can actually get a rescue of a non-productive VJ alpha join. So you can get, if you have an initial non-productive rearrangement, then you can get a subsequent rearrangement that bypasses that non-functional gene to hopefully result in one that will work. Small point to remember. Okay, so this is maybe a little bit out of context, but I wanted to leave this graph in because this was a very confusing point to me when I was studying for the boards, but there are two different ways to look at flow cytometry. So one of them is where you see a graph and you will see a line with peaks. And we see this a lot when you're talking about
CGD.
CGD, thank you. So you're supposed to be able to tell if it is autosomal recessive CGD or X-linked CGD, right? And when that's happening, what you're doing is you're drawing it in a way that you've looked at a marker. And in the case of CGD, the marker you're looking at is something that it tells you if there's an oxidative burst. So I think of flow cytometry, everything is binary, yes or no. So is there oxidative burst or is there not oxidative burst? It's not exactly true, but. So in that case, think of a bunch of test tubes all lined up in a row. Okay, and then say if it says yes, there is a oxidative burst, you're going to drop a ping pong ball into that test tube. If there's not, you can drop it into a different test tube. So then what you're going to see is these peaks building as you put in these ping pong balls. And it's basically just going to tell you, yes, there is that oxidative burst, or no, there's not. You could do the same thing and ask for CD4. So you always have to pay attention to your axes and see what you're looking at. So that type of histogram, if you saw CD4, you'd just be popping ping pong balls into into a test tube over here, say every time there's a CD4, and if it doesn't see it, you're going to put it here. So you probably have 2 peaks about equal size. That if it doesn't have CD4, then well, it's probably CD8, I guess. Okay, but there's another way to look at it, and that's what these are. So, right, this is a scatter plot. So instead of dropping into test tubes every time you see something, every time the computer sees something, you're just going to draw a dot on this map. Okay, so each one of these dots represents a cell. So it's almost like looking at the same thing from above. So if it has both CD4 and CD8, it's going to drop a dot here. If it has CD4 and not CD8, it's going to drop a dot down here. If it has CD8 and not CD4, it's going to drop a dot down here. Okay. So here, of course, it starts with double thymocytes, which we talked about. So it's going to drop all the dots here because it's expressing both CD4 and CD8. And then it's actually going to change, and most of them are going to become either CD4 expressing or CD8 expressing. So it's going to drop a dot here. CD8, yes, but CD4 is low. CD8 is low up here, but CD4 is positive. So these are CD4 positive, these are CD8 positive. It's kind of a silly point, but I really struggled getting through that because every time I looked like flow, it said it looked like it was right saying something different. But these are the 2 main graphs you're going to see on the test. Either these scatter graphs, and you just have to look here and see what it's looking at. So if the cells are over here, it's high in this, and if they're over here, it's high in this. It's high over here, it's high in both. Well, both. Or it's going to be those histograms where it's just dropping balls in the test tubes, and you're going to see peaks. And it's going to be one thing you're looking at, not 2. Okay. So once T cells have progressed to becoming immature T cells or mature thymocytes, right, they undergo both positive and negative selection because they're MHC restricted. So the T cell has to be able to recognize your own MHC in order for it to survive. So that's the process of positive selection, in that each of these T cells will look at your MHC, and if it recognizes it with just low avidity, then those T cells can actually continue on. They're good. So they've been positively selective. They've been allowed to live. But then we know it can recognize your MHC, but then if it recognizes your MHC with self, then they're going to be negative selective. Those are autoreactive. You don't want those. You want those killed off. So those are negatively selected at that point. And then only the ones that recognize MHC with low avidity and then recognize your MHC with, do not recognize MHC with self-protein loaded are going to become mature peripheral T cells, mature naive peripheral T cells. Okay, so it was a question for a long time about, okay, well, how does this negative selection occur? If you're in the thymus, how do you even know whether you have, right, if you're self-reactive to cardiac tissue or to skin tissue or what have you? Well, then they found this gene called AIR, autoimmune regulator gene. And what it seems to do is be able to cause expression of low levels of different self-antigen in the thymus. So we do think that T cells can see all your different self-tissue in the thymus because of regulation by this AIR gene. And we know now that some people have a defect in AIR gene. If they do, they have something called autoimmune polyendocrinopathy, candidiasis, ectodermal dysplasia, or APOSED, or they have autoimmune polyglandular syndrome type 1. So if they aren't getting expression in the thymus of all these different self-tissues, then it's not getting properly negatively selected. So autoreactive T cells are actually going to get through, and then you're going to probably end up with multiple autoimmune problems. Okay. That's about it for T cells. So once these cells, these lymphocytes get out to the periphery, well, where do they want to go? Well, they've gone through the central lymphoid tissue, and now they want to go to the peripheral lymphoid tissue, and this is probably where they're going to come into contact, right, with antigen. So to get there, there's a sophisticated series of signaling events that need to happen. So, right, they go through the vessels, and then as the vessels come out into these lymph nodes, they turn into something called high endothelial venules, HEVs, where the lymphocytes are able to sneak out. And once they're able to sneak out, then they're able to get into the T cells. They follow into the lymph nodes this gradient called the CYP1 gradient, which I would know. So I think the CYP1 gradient increases. You have the CYP1 receptors, so it's following it, it's sniffing it out. And then to get back out, it starts losing those receptors, and the gradient decreases, and they're able to sneak out. Sneaking in, they follow the CYP1 gradient. They escape through these high endothelial venules. And then if they express something called CCR7, which the T cells would do, they interact with CCL19 and CCL21, and they go to the T cell zone. B cells, on the other hand, are going to express something called CXCR5, and they're going to interact with CXCL13. And that's going to bring B cells to the B cell zone. But as these get activated, these aren't good markers for T cells or B cells, because say as T cells get activated, where do they want to go? Well, they want to end up going to the B cell zone so they can actually activate a B cell, right? So then CCR7 is going to be downregulated, and CXCR5 is going to be upregulated, and it's going to let that T cell move towards the B cell zone of the lymph node. In general, I would definitely know all four of these things: HEV, CCR7, CXCR5, and CYP1. The CYP1 gradient, or be familiar with it. Okay, so right, the chapter goes back to B cells for just a second. A couple things that I picked out that I thought were important. So one, B cell survival in the periphery is dependent on these two cytokines called BAFF and APRIL, which interact with receptors BAFF receptor, BCMA, and TACI. And similarly, this is just, again, kind of another random point, that you have your traditional B cells, right? But then just like you have these gamma delta T cells, then you have other B cells that are much less diverse, like marginal zone B cells and B1 cells. So marginal zone B cells are found in the spleen. The B1 cells or B2 cells are found in the body cavities like peritoneum, pleural, secondary lymphoid organs. Oh, I'm sorry, B2s are conventional B2. B1 are the ones that are not very diverse, and marginal zones are not very diverse. They mostly secrete IgM, which is called a natural antibody. Again, not very diverse, but even someone who seemingly had no B cells in their blood, you may sometimes see some IgM, be able to pick up some IgM, which I usually think is just natural antibody, which is mostly directed at carbohydrate antigen. Okay. And just for review, at the very end, there are two kind of summary slides of B cell and T cell development, of course. So B cell, starting with the stem cell, going through rearrangement, all of which we've talked about before, until it becomes a mature, naive B cell. Out in the environment, can interact with antigen and either become a plasma cell or memory cell. And then there's one similar for T cells. I think these are very nice. It doesn't have everything you need, but it does help you know what's on the outside of these cells at different stages. Do I think you need to know everything? No. Most of it, yes. I probably know at what stages CD19, CD20, CD21 are expressed. And obviously you should know when the recombination proteins are being expressed in both cases. When things are double positive, double negative, single negative, or single positive. And that is it. All right. So my takeaway from this chapter, I found it very dense and hard to get through. Trying to know these big, broad concepts. When you need to get down to memorizing, memorize from the tables. Don't remember the mouse stuff that was discussed. I don't think that's very important to study from. And that's it. Any questions?
So at what point during the life of the individual do each of these processes occur?
Well, it's probably actually a good thing to know as well, right?
Well, I don't know how much of it is really known.
Yeah.
You know, I mean, I've heard arguments about it for years and years and years, but, you know, I mean, has it all occurred by the time you're born? Or is a little bit of it still going on? Or as an adult, do you actually have the ability to do things like this? I mean, can you actually rearrange and recognize some self that you had failed to recognize?
Mm-hmm.
Or, you know, can you, I mean, you certainly can lose the ability to recognize self or
Mm-hmm.
fail that, and that sort of fails. And so that's for self-recognition. And then what about foreign recognition? That's looking at the puffs here.
Oh, yeah.
You know, at what stage of the life of the individual does foreign recognition start to occur, or foreign tolerance start?
Or oral tolerance, yeah.
Or oral tolerance.
Perfectly, yeah. That's a good point.
Well,
those are all questions that you guys probably hear the answer.
And laugh at us for not knowing.
interesting. Dr. Murkart, who we just talked about earlier, is enrolling people in a study for transplants, on which she's actually doing this. Not only is she transplanting solid organs, but she's taking a piece of the donor thymus and transplanting that as well, hoping that decreases rejection.
Like with the other solid organs.
With the other solid organs.
That's really interesting.
The kidney transplant people used to give a blood transfusion from the, you know, if it was an ABO compatibility, they would give a blood transfusion before they gave the kidney. I don't know what the exact
From the same donor, right?
Yeah, I don't know if they still do that or not.
I thought they were looking at that, and then my recollection was there was a setback, but I don't remember what it was. But initially, I think it even made the New England Journal that there was some really huge success, and a couple of people were off rejection meds, and then, is that true? Wow. Yeah, I don't remember. I may be wrong about that. I don't remember.
Yeah, I mean,